the fine control of electrical circuits with the complexity of the fluxes in the different sections of the plant. And this connection between these two areas allow us to have the precision and control of the plants that are running nowadays. So the idea of this connection between synthetic biology and metabolic engineering is precisely to be able to combine these two things, synthetic biology and metabolic engineering, to bring this fine control or tunability of the metabolic pathways in order to do with these pathways what we need to do to get applications out of them. And the final goal of all this is to be able to move our society towards the bioeconomy. So this is a bio-based economy where everything that we build, that we need, and we eat is made in an eco-friendly manner, biologically based way. So we are trying to move away of the fossil fuels and petroleum to make all our materials and so on. And one way to do that is microbial biotechnology, or using microbes to produce all these compounds, chemical compounds that can substitute all the things that we make from petroleum, like biofuels, chemicals, materials. And there are many organisms out there that are able to produce these things, but they normally produce them in very tiny amounts, which makes them very expensive to produce. That's the big constraint. But luckily we have synthetic biology and metabolic engineering to be able to engineer these organisms to maximize the production, to reduce the cost of the substrate they use, and so on. So it makes the process economically feasible. And we are seeing a lot of examples in the last years how these bio-based technologies are able to actually build products in the market that can compete effectively with petroleum. So I'm going to go more in detail into this design, build, test, learn cycle that you all know because it's pretty popular in the synthetic biology field. And I'm going to use this as an example to show you how we, in our lab, engineer strains to make them better in each cycle until we reach the point where the strain is good enough to be able to reach the market or production level. And I also going to use this opportunity to introduce you to some of the techniques that my lab is using in a daily basis. So everything starts with the design phase. Basically, the researcher is in front of the computer and use the literature, research, and models, mathematical models that can be based on like genomic scale models or ODE models to simulate what happened when you do the engineering of the organism. And that predicts targets that then we're going to build in the lab. And to build them, we're going to need to make put the pieces of DNA together using DNA assembly techniques such as Golden Gate, and then we're going to introduce them into the genome of the organism using other techniques like CRISPR-Cas9 that I'm sure you're all familiar with. In addition to that, we also do evolutionary engineering or the introduction of genetic circuits, more complex circuits. And then when we have the strain that has been built according to the design, we test this strain at different scales from microplates to big bioreactors, and we optimize conditions, and we analyze using GC, HPLC, and other analytical techniques how they behave, how much they produce of the compounds we want them to produce. And then this is normally the case that what you expect is not always what you designed. And this is an opportunity to learn why this is happening and how we can further improve these strains. And this is the learn phase that is basically the utilization of different systems biology tools, such as transcriptomics, proteomics, and metabolomics, and can integrate machine learning as well to try to find new targets for the second round of design. Another very important steps for designing bioprocesses is the selection of the microbial host. So there's many organisms out there that can be selected for your bioproduction approach. And selecting which one is not always obvious. So there are two main let's say, groups of organisms, or way to separate these groups. So one is the so-called non-conventional organisms, and the other one is the conventional organisms. So as you can imagine, conventional organisms are those where we have a lot of tools and knowledge about them, like E. coli, Saccharomyces cerevisiae, where we have like Golden Gate libraries, we have CRISPR working efficiently, we have genome scale metabolic models, we have a lot of biochemical knowledge on the metabolism of enzymes characterized, and so on. But they have a disadvantage that sometimes they are quite far away from what you want to achieve. So maybe like Cerevisia, we all know it's very good at producing ethanol. It has been used for many years to make like beer, wine. It might not be very good to produce lipids, for example. And trying to convince genome of Cerevisia to make something radically different is going to be a huge task. And maybe going to something else that is more closer or naturally produce the precursor of the things that you want, or even the things that you want, it can save a lot of time and energy. And here's the non-conventional organism come in place, because and this is like 99% of them, right, are those that are more limited in the amount of knowledge that we have on the tools that we have, but they could have a big potential to be selected for bioproduction, because they might be already producing the compound or the precursor of it. So, and the good things is that now with synthetic biology, it's very, very easy. But it's something easier than before, at least to convert these non-conventional organisms into conventional ones by using synthetic biology tools and going through this domestication process. So now it's very easy to sequence a whole genome of a new organism. It's very easy to do a transcriptome RNA-seq analysis and identify strong promoters. It's easier to develop gene editing tools thanks to CRISPR, and so on and so forth. And this is an example of the organism that my lab uses. And you can see it's mainly yeast. You have here Saccharomyces cerevisiae, Gerobia politica, probably the two we work more with. But we also work with bacteria, with E. coli, subtilis, and for specific purposes, we use specific bags. So I think this is the right philosophy for my group. Like, instead of trying to rely in the model organism to do everything, try to think first what is needed to produce and what will be the best organism to do it. And my lab has three different research lines that are somehow connected between them. And I'm going today to tell you examples of each of them, at least. And the three lines are the developing of synthetic biology tools here on the left. Many of them will be intended to two metabolic pathways in a better and a more efficient way. We also work in the second line that is here in blue, the metabolic engineering or the engineering bioproduction in these microbes to make specific products. And finally, we're also working in a line that is about creating microbial communities that can have application in biotechnology, but also in biomedicine. So I'm going to start for the one in between, the bioproduction, the one that I basically introduced in the past few minutes. And regarding bioproduction with microbes, something that I didn't mention and I think is worth saying is that when we think about producing something, it's not only about making a lot of the product, like producing a lot of titer, a lot of gram per liters of it. It's also about the economy of the whole process. And the process is not only producing something in the bioreactor, but also how it is produced or what it is produced from. So the upstream thing, the things that, for example, the substrate that need to be introduced into the bioreactor. So the cost of this substrate can really make a big difference. Or the downstream part of the process. So once you have produced a lot of this compound, it need to be polished, it need to be isolated, it need to be purified, and that can be also very expensive. So when we design our strains, we maybe can use synthetic biology also to bring to the strain features that help to make the overall process cheaper. So not only producing a lot, but also be able to produce a lot from a very cheap carbon source, and also be able to purify this product in the end in an easier way. So this is just a small summary of some of the projects that we have done in the last year, so you can have an idea of the kind of things I've been doing in this bioproduction field. So with this filamentous fungus as Aspergillus, we have been producing different vitamins such as riboflavin or folic acid. We've been producing lipid-derived compounds like the fish oil that Minos mentioned in the introduction, or biodiesel. We've also been producing flavor enhancers like guanosine and inosine. In Gerobia politica, which is a filamentous, is a yeast that accumulates a lot of lipids. We've been producing lipid-derived chemicals with specific purposes, but also organic acid that can be used as a building block for the chemical industry. We've been also producing enzymes and proteins, but also aromas and fragrances, and terpenoids and flavonoids. And on top of that, we've been also engineering Gerobia lipolytica to be able to produce these compounds using low-cost carbon substrates. So, for example, starch or cellulosic materials. But I'm going to talk about this in a minute. So I selected two examples to guide you through, so you can have a better understanding of how these kind of projects are started and are completed. So one is an illustration of how synthetic biology tools, and specifically this build part of the DBTL cycle, can help to produce beta carotene. And then I'm going to talk about another example, that is how we can engineer this host to use this low-cost substrate to reduce the overall cost of the production. So the importance of beta carotene is, you've probably come across this compound and read things, because it's quite abundantly used in synthetic biology research, because it has a color, so it can serve as a tool to visualize the production of a metabolite, which is great for many applications. But in our case, we were not only interested in using it as a tool to understand better pathway, but we actually wanted to produce a lot of this compound, because this is very interesting for the industry. There is a high need to make it. Most of it comes from petroleum at the moment. Some of it is produced by algae, but it's really a minor part. And the beta carotene is used because it's an antioxidant, it's a precursor of vitamin A, and it's a colorant. So it's used in beverage and food industry a lot. It's used in cosmetics a lot. It's used in food supplements and many other things. So the thing is that Gerobia is not able to produce beta carotene by itself, because it lacks these genes here in red. So it has a mevalonate pathway. It can produce GGPP, but it cannot go further. So it needs to introduce these genes from other organisms that are able to do it. But the reason why I selected Gerobia, and that links back what I was explaining before about selecting the right host, is very important. We selected Gerobia because Gerobia is very well known for being able to produce lipids, and lipids comes from acetyl-CoA, which is a metabolite that is also the precursor of the mevalonate pathway. So we are assuming that the acetyl-CoA pool in Gerobia is quite high, and that will allow us to produce a lot of beta carotene. So in order to make this project, we had to develop a new tool that was not available at that time for Gerobia lipolytica, which was Golden Gate. I'm sure many of you are familiar with Golden Gate, but basically what you need to know to understand the rest of the talk is that it's a DNA assembly method that is based in restriction enzymes type 2S, which means that a type of restriction enzyme where you can program the overhangs, like so you can really create these libraries like this one here, where you have a lot of like promoters part, genes part, terminators part, markers part, integration site, and they are designed in a way where they are compatible in a specific order, so you can make this cassette very quickly, and you can make variants also very quickly. This is a method that I particularly like for metabolic engineering approaches. So we developed this library for Gerobia, and this allows us to make this cassette that you can see here on the top right. That is a cassette that was expressing 3 genes. There's 3 genes here. So the 2 ones that need to come from heterologous sources, but also the last one in the pathway that we thought could be a limitation. And we built this using the Golden Gate, and we introduced this into the genome of Gerobia lipolytica. And we introduced this into 2 different Gerobia lipolytica strains. One is called wild type. That is the one that is it's a modification of the wild type that has a couple of auxotrophies that allow us to introduce heterologous pathways. But we also use the one that is called obese, OB strains, that basically was engineered to accumulate more lipids. So when we introduced this cassette into Gerobia, we could see quickly the change in color. And I forget to mention, but beta carotene has an orange color, and actually is the responsible for the orange color in carrots, for example. So it was good. We started to see this yellow color, orange color. That means beta carotene is being produced. And something that was surprising is that the obese strain, the one that produces more lipid, is more orange. And you can see here the quantification of the beta carotene is higher than in the wild type. And I'm saying this is surprising. This was surprising because, as I said, the two pathways, lipid pathways and mevalonate pathways, they share acetyl-CoA as a precursor. So what I was expecting at the beginning was if you have more lipids, you have less acetyl-CoA available to make beta carotene. And that was not the case. And we're still trying to understand why this is the case. But one theory that we have is this picture here on the bottom left, which is a cell of Gerobia. These in green are the lipids. And these things in orange is the beta carotene. And you can see they colocalized, which means the beta carotene, which is an hydrophobic molecule, is dissolved preferentially in these lipid bodies. And this helps solubilizing and storing this beta carotene, which otherwise will go to the plasma membrane and could produce some toxicity. So because you have more lipids, you can store them and they don't interfere with the cellular processes. So that was good news. We were able to produce beta carotene, and we wanted to produce more to be able to reach titers that could be potentially applied in industry. And something that we knew from research in other organisms like Carmen cerevisiae is that HMG1 gene, one of the genes in the mevalonate pathway, is tightly regulated. So if you overexpress it, a truncated version of it that hasn't had feedback regulation, it normally increases the flux in this pathway. So we did that. And we see this is a strain that I mentioned before, the obese strain with the carotenoid cassette, which has these three genes. And in that, we introduces the HMG1 overexpression gene, and this increased by 2 times the production of this compound. And then we introduced a second copy of this cassette. And the second copy of this cassette also increased like 20 or 30% more the production of the beta carotene. So what this was telling us is that maybe we have a third copy, we can produce even more. Maybe we haven't reached the limit of production of these genes to maximize the pathway. So we wanted to investigate where this limit was. And for this, we took advantage of this golden gate system that I briefly introduced. And one of the nice feature of this assembly method is that you can generate these combinatorial libraries. And this is what we did. We did a combinatorial libraries where each of the 3 genes in the cassette could be controlled by each of the different promoters that we tested. So in the end, when you do the assembly, like because all the promoters share the same overhangs, they all can go randomly in this position. So you will have a combination of all the possible cassettes where all the genes are controlled for all the promoters. And because in this case, these compounds can be easily screened by color, we can transform Gerobia with the pool of cassettes and select which were the most orange one, and then we could sequence them and see which are the promoters that were producing the most orange gene, the more orange strains. So we did that, sorry, and we found something like that. So different colors. And we picked the most orange ones and we sequenced them, and this is what we obtained. And we found that the one that produced more beta carotene was the one that had the 3 strongest promoter controlling, so the same strong promoter controlling the 3 genes, which was the strongest promoter. So what we did next is to take the strain that we presented before with this cassette of the carotenoids and also the HMG1. We introduced a second copy of the cassette that we identified as the best one. And this increased that we saw before with this cassette. And then we introduced a second copy of this cassette. And we found that there was little increase now, which means even if we go further, if we improve this expression even better, we are not going to see, because now the bottleneck is somewhere else in the pathway. So with this, we had a strain that was quite orange, with producing a lot in flask, but we wanted to try like more real, larger scale conditions. So we went to the bioreactors, And we optimized a little bit bioreactor conditions, and we managed to produce 6 grams per liter of these compounds, and you can see the cells with all the beta-carotene inside. And that's the highest production that has ever been described of this compound so far. So that was for the beta-carotene production. And now, we're going to see how these lipids of these compounds can be produced in a cheaper way by using low-cost substrates. And the low-cost substrate that everybody wants to use, because it's very abundant, it's very cheap, is lignocellulosic materials. So all the waste biomass that is generated in forestry, in agriculture, and so on. And basically these lignocellulosic materials can be hydrolyzed to their main components that are glucose and xylose. Basically, glucose can be used by Gerobia very nicely. All the previous experiment that I showed for the beta-carotene, it's using glucose, but Gerobia cannot use xylose, which means the conversion of the substrate into products is going to be very low because xylose wouldn't be used. And then the goal of this was to engineer Gerobia to use xylose and to produce lipids out of xylose first, but then go to the real product, which is the lignocellulosic substrate. So we looked at the Gerobia genome, and it has the genes. It has genes that can metabolize xylose in theory, by homology, like the xylose reductase, xylitol dehydrogenase, and cellulose kinase, that convert xylose into intermediate terpentophosphate pathway. But Gerobia was unable to grow, so we did some expression analysis, and the expression was very low. So we decided to overexpress these genes, and we overexpressed these 3 genes using a strong constitutive promoter, and Gerobia was suddenly able to grow in xylose as good as it normally grows in glucose. And we use this using the native genes, but also heterologous genes from organisms that are well known to use xylose. So that was good news, but we wanted to try the real thing, as I said, the lignocellulosic hydrolysate. And we did this in collaboration with a Mexican group. I was seeing a question, but I think I can answer the question in the end better. Yeah, and now I cannot read off the question. Sorry. Yeah, sorry, I was getting this question, but I will answer to it later, and I was trying to fix it in my screen because I couldn't see my presentation anymore. Yeah, sorry. So yeah, we partnered this Mexican team, and they were using agave hydrolysate. Agave is the plant that you can see here, and this pine here is used to make tequila. So for every liter of tequila that is made, 10 kilograms of biomass is wasted. And there's no use for this. It's basically accumulated in the lands for many years until it's degraded. But it's still rich in sugar, so we wanted to use this. So we use a strain that can consume xylose and can produce lipids. And we wanted to, well, we tested it in agave hydrolysate. And we found this. We found that Gerobia is able to grow in different concentration of the hydrolysate. And the more hydrolysate you add, the more Gerobia grows. This can sound like obvious, but at the same time, it's very interesting because this is not the case with most conventional organisms. If you put Saccharomyces cerevisiae with hydrolysate, it will not grow in high concentrations because when you extract the sugars from the lignocellulosic materials, you also release other compounds like phenolic compounds and phosphorus and also acetate that basically can kill your cells. But Gerobia was very, very good at coping with these toxic compounds and even was able to use them to grow. So it was one of the other examples of good things of using non-conventional organisms. So well, we have Gerobia that was able to use xylose and was also able to use lignocellulosic hydrolysate. But we wanted to maximize the conversion of the substrate into the product. And in order to do that, we expressed an heterologous pathway to connect the use of xylose to the generation of the precursor acetyl-CoA to make lipids. And for that, we tried these 2 different alternative pathways, and we found that both of them work really well, and they increased the lipid production in 60 or 40%. So, and then we selected the best of these conditions and strains, and we optimized the production in bioreactor. And you can see here in the image how a lot of lipids were produced from lignocellulose, from very, very cheap lignocellulosic hydrolysate. And we actually achieved the maximum theoretical yield of production. Okay, I think we're good on time. I'm not sure if there was a limit on time, but I think I have a couple of more things to present, but I think they're going to be shorter than before. So this is a brief introduction to the other two research lines that I'm working on. So one I mentioned already, it was the generation of these synthetic biology tools, and more concrete, I'm working in multiplexing Crispr. So you're probably familiar with Crispr as a genome editing tools and transcription regulation tool. So the word multiplexing basically is telling us that CRISPR allow us to basically target many different targets at the same time. And this is something that was more difficult to do in one go with previous techniques. So this is I think an ideal system for the coming metabolic engineering and biology approaches. And that's why we're trying to optimize these methodologies. So we have developed this orthogonal CRISPR system that is programmable. So as you know, different guides will target different things, and you can have different repression domains to downregulate the expression of certain genes. But the key feature is that you can multiplex that, as I said, by expressing multiple of these guide RNAs at the same time. But the problem that we found when we wanted to use this is that it's not trivial to express a lot of different guide RNAs at the same time, because they have a lot of repetitive domains. They are difficult to clone together. Because of that, you cannot order synthetic blocks, DNA blocks with this, because chemically they are difficult to make. So we came up with a project where we developed an assembly method based on Golden Gate as well, to be able to generate these guide RNA arrays, where we are able to produce more than 24 guide RNAs in one go. That means we can potentially target 24 different genes at the same time in one single transformation, which really speed up how we can engineer strains in the future. Another thing we are working on, and it's related to the same project, is in the future we want to move to a total automation of strain development. So we envision a future where you just type in your computer what is the product you want to make and what is the organism you want to use, and then automatic design will tell you what are the targets you want to find and what are the guides you need to use for those targets, and then it basically constructs for you this array, guide RNA array that you build with Golden Gate system we developed, and you just need to transform the strains to be able to modify in a highly multiplexed way one strain. And we tested this for an example like succinic acid production, and we found how expressing 12 or 24 guide RNAs in one single transformation allowed to multiply by 7 times the production of this compound. The last example or topic I'm talking about is the use of synthetic microbial communities. And I believe this is also the future of this research field of bioproduction. And there's a lot of synthetic biology going on in this area as well, as microbial communities, as you know, are very relevant for human health as a gut microbiome or skin microbiome, but also for plant health through plant microbiome interaction like nitrogen fixation. It can be used also for bioremediation, and we're starting to apply things to use it in bioproduction. And these things here on the right is just hypothetical future bioreactor where we will be able to design each strain to make one thing there and to fully use substrates to maximize the conversion of them into products, to minimize waste, engineer recycling, and to be able to tune what we want to produce according to what inducer do we have. So something more similar to what happened in nature, where there's never a single organism doing something, there is always a community that has evolved to make something much better. And we are exploring concepts like division of labor, cross-feeding, and population control, and we are developing tools to allow this to happen. And there are two kind of communities that we are working on. One are called like fully synthetic communities, and it's basically when you design from scratch the community you want to build, the connections you want to have, how do you want things to relate to each other, to exchange nutrients, to exchange signals, and you build these functionalities using synthetic biology. The other example, and that would be an example, that would be something similar to what I presented here when you create a bioprocess that is fully controlled. The other example is what I call semi-synthetic microbial communities, where you take an existing community, like it can be the gut community, but also can be a community that makes wine, for example. And you can take one strain out, you can engineer that strain with a specific feature that you want in the community, you put it back in the community, and you have this semi-synthetic community that now have an additional feature. And the example I selected from the project I'm running at the moment in this area is a semi-synthetic microbial community one, where we work with soy sauce production. So soy sauce is basically made by a double fermentation by a microbial community. The first one is basically the first fermentation is made by Aspergillus and taking the soybean biomass and converting it into amino acids and sugars. And then there is a second fermentation where there is a microbiome, a complex microbiome made of a lot of bacteria that are able to convert all these nutrients into soy sauce. But the problem, or not, but well, it could be considered the problem of the aim of the objective of the work was to try to make soy sauce that were not black. It's not like black, it's bad for health or anything. It's just that there is a high interest, especially for high cuisine, to make something with the strong flavor as a soy sauce, but without the dark color that basically change the color of the whole dish that you're cooking. You can actually buy clear soy sauce now in Amazon. It's like super expensive, and it's basically generated by using different columns to remove the dark color, but it doesn't remove only the dark color, it also removes most of the flavor. So we thought using synthetic biology, we could make clear soy sauce, but using but not using chemicals to remove things and keeping all the flavors. So we identified the community that was naturally able to do this process, and we identified some of the members of them, that was Bacillus subtilis, that are bacteria, that gram-positive bacteria that we know how to engineer, and we engineered them to basically fight against the generation of the dark color. The dark color is basically melanoidins, which are these guys here in the figure, that are naturally generated through the Maillard reaction that happen spontaneously between amino acid and sugars, and specifically xylose. So our strategy was to change the community to reduce the amount of xylose by converting to xylitol, so there's less xylose available to make melanoidins, but also expressing enzymes that can degrade these complexes. And we did that. So we generated one type of strain that this one job, the other strain that did the other job, and we tested them independently, and we test a community where the two strains were together, and we found that the browning color, or the dark color, was reduced from the wild type to the one that one single modification, one with the other single modification, and in blue with the two modifications at the same time, with the co-culture of the two strains that reduced this browning to more than 95%. And with that, I'm done for today. If you're interested in knowing what other things we are doing, you can go to our website, rlalab.org. And yeah, before finishing, of course, I want to acknowledge all the team and our collaborators and the fundings, because without all the members of the group, it would be impossible to do all this research. And just before the last slide, saying thank you, I take this opportunity to say that I'm going to have some postdoc position available soon. So if you're looking for a postdoc, please, and you're interested in this, contact me. Yeah, thank you all of you for your attention. I will be very happy to answer any question.
Thank you very much, Rodrigo. This was really, really great and fascinating, and so many topics to go through. So we have the first question in Q&A. Arin, would you like to ask your question? And maybe Rodrigo wants to answer that. Okay, so maybe Rodrigo, you can just have a go on. So first one was, yeah, just go for it.
So the first question is, have you tried to quantify the contribution of each cassette with different promoter strength to overall beta-carotene production? I don't know if this was asked before I showed all the combinatorial library approach. I can kind of answer that question. If you talk about...
Yeah, it might have been before that. You go back to that slide, please.
Sure.
Thanks.
Yeah, I don't know.
Okay, the combinatorial. And so I was asked specifically about, I think it was 2 slides later. I think the slide after that. Yeah, that the bar chart over there. Have you? I think I was thinking of like maybe looking at how much like each of the 3 types of promoters contribute to the overall beta carotene content.
No, so we didn't really. That's something we were expecting to find actually different promoters. But what we found is the strongest promoter was the best one when you put in front of all the genes. So that, I think that means that in this case, that probably there is no clear bottleneck in one of them. It's not like we found a lot where one single strong promoter would be enough.
Okay. So the main thing is like the main thing you've seen is that it's more of a combinatorial thing rather than like having just one strong promoter does everything.
So in that
is that the main takeaway?
Well, the main takeaway is like depends on the pathway, and there will be pathways where one gene is the expression of one gene will be the bottleneck, and maybe expressing that the maximum, and the other one the minimum will be the best. So sometimes when you express a lot of enzymes a lot, you also generate some burden to the cell. But that was not the particular pathway. In this particular pathway, the most we express, the most we produce. So the take-home message, I think, is you need to, the combinatorial assembly can serve very good to identify if your pathway have a bottleneck or not, and also to coordinate production with metabolic burden.
So that's like this little segue to my second question about the burden. And I think you've already answered the question. So maybe.
Yeah, but
maybe there is a burden issue in there.
So we don't observe any burden, and this is something that this is the case most of the time in Gerobia. This organism in particular, we don't observe a lot of burden when we express.
Okay.
And the genome of Gerobia is significantly larger than others like Cerevisiae. And I think there is more flexibility for the proteome to adapt to the heterologous expression of genes. So we normally don't observe metabolic burden for the expression of genes. We can observe growth defect when we express something that messes up with metabolism, but not for the expression itself normally.
Okay, this is interesting. Thanks a lot.
Okay.
Thank you. Reza?
Hi, thank you so much for the great talk. You emphasized the importance of kind of keeping the techno-economic feasibility of your process in mind when you're designing your project in the beginning. And I was just wondering if you have, or what process you follow to do that, or if there is a formal process, or if you're mostly relying on just your own experience and knowledge of the field, because that's something I'd quite like to do within my own project. But I don't really know where to go about figuring out, what substrates would be the correct ones to use, whether the unit economics of the value of your product at the end makes it worth it, the different types of fermentation processes, and so on and so forth.
Yeah, that's a good question. And I would say there are 2 things. So my experience tells me is that the cheapest you can go, the better. So you always try to reduce cost as much as you can, because normally, yeah, the cheaper it is, the most likely it's going to end up in the market. And even if end up in the market and you can make it even cheaper, you're going to have larger margins. So you will get more money. So everybody happier. But yeah, there's a formal way to assess, and there's just the techno-economic analysis, life cycle analysis, that can go all the way from the selection of the substrate to the process itself, different ways to make the process, the purification, and it can go all the way to the distribution of the product to the final customer and things like that, packaging and so on. And that's taking into account both the economy of the process, but also the environmental impact of the process, how much CO2 you are producing and things like that. So yeah, I mean, I don't think there is like a standard way go through kind of things where you analyze all these things. It's kind of complex, but yeah, I would suggest you to read about life cycles and assessments and these kind of things.
Great, thank you very much.
There's another thing with this is like, it's very difficult you really need to understand the market in the specific region where you want it to be produced, because the substrates, for example, can change a lot. Like if you want to produce this in a country where you have a lot of sugarcane, for example, sugarcane is the best substrate. If you want to produce someone from sugarcane in a country where there's no sugarcane plants and you need to ship it from Brazil, that's going to be super expensive.
Right, I see, yes. Very interesting, even more complicated then. Thank you.
Thank you, Reza. Jose?
Hi, can you hear me now?
Yes.
Yeah.
I've read a lot about different projects, such as engineering microbial communities to clean up oil spills, or engineering microorganisms to capture carbon. Obviously, there's a lot of downsides, and the negative side is that it's really hard to establish stable microbial communities in already soils that have other communities. So I was wondering whether you're optimistic about certain areas or projects in tackling climate change through metabolic engineering.
Well, I'm in general very optimistic about everything. So I guess that can be biased in my answer as well. But yeah, I'm optimistic. I really think strain engineering in the biology can have a lot of potential to help many of the things we are doing wrong at the moment. And I mean, we haven't invested much compared to what has been invested in the oil industry, for example, right at the beginning of the oil industry. So I think there's a lot of things that we need to do, but there's a lot of things that can be done. And we are, for example, I think the synthetic communities kind of area is relatively new, and there's a lot of things that we are learning now. So hopefully in a few years' time, we will be in a better place to really engineer these natural communities. And there's I think a big limitation at the moment, that is regulation, that is preventing also the field to progress faster, right? When there's no much funding that goes to projects where you need to create GMOs that at the moment cannot be applied in fields. So I guess we need to go in parallel with the technology and the regulations to try to solve these problems. But I really think that we can do it.
Thank you.
Okay. Reine?
Hello.
Hello.
Thanks for the presentation. I have a question. It's a general question actually about the bacterial community. Usually when bacteria wants to uptake carbon sources, if sugars are abundant in the media, they prefer to take the sugars rather than the lipidic carbon sources. So is it a way to somehow manipulate the bacteria and force them to uptake lipids sooner than the sugars, even though when the sugars are abundant in the environment.
Yeah, yeah. I think there are ways. We are working in these kind of things as well. And so one of the things you can, of course, if you know how the mechanism work, the metabolic repression work, you can always engineer that not to happen in your strain or in one of the members of the community. If you don't know how the mechanism work, which is often the case, especially with non-conventional organisms, you can always rely on evolution. And if you have a substrate there that is not being used, and you can try to make it consume a little bit, and you evolve that community, it will probably evolve to consume that, because normally, especially in substrate consumption, all the machineries are normally there.
So I guess you have to somehow stop this catabolite repression by manipulating some of the genes, yes?
Yeah. So you can manipulate, if you know how the mechanisms work, if it's so you know the signal cascade, for example, or you know what is the transcription factor that do this catabolite repression, you can remove this transcription factor.
So you said that you did some works on this. Did you work specifically on lipid uptake, the fatty acid uptake by the bacteria, or?
So we are nothing in particular with thin lipids and sugar, and this is because we never found a system where lipids are you thinking on a specific substrate where sugars and lipids are abundant?
Yeah, what I'm doing now is in the wastewater treatment, the microbial community, they when these sugars are abundant, they uptake them, but they don't degrade the lipids. So I came across this, that one reason can be this catabolite repression, that they usually express some genes which stop the lipase production, and then the lipids remain undegraded in the system. So I was thinking if it's a way that we can manipulate and stop this catabolite repression, we can also improve this wastewater treatment.
Yeah, definitely. Yeah, definitely. And if you know it's only a lipase, and you can test this very quickly, just adding an external lipase, that will be as simple as expressing a lipase heterologous constitutively, and then that will be done. If the problem is the consumption inside the cell, you can always overexpress things like beta oxidation in the bacteria, so you will have always active the degradation of the lipids. Yeah, there's ways, and I'm happy to talk more about this if you want in a different time.
Yeah, I can maybe email you later. And just one last thing, is it economically feasible to go for such a processes to manipulate the bacteria and then add them to the wastewater systems to see if they are working or not, or still it's not really economical to do this kind of things?
Well, in this case, I think it's economical because there is no much cost, right? You just need to substitute one of the organisms or modify one of them, and the process will be the same, and the process is running. So you just make the process more efficient. It will be economical. The problem maybe is if it's allowed, depending what the restriction of the legislation to use GMOs is.
Yeah. Okay. Thank you very much. Thank you.
Thank you. Yafeng?
Thank you very much for the nice talk. My question is, when researchers try to improve the production of terpenoids, they mainly focus on the MEP or MVA pathway. And do you think it's a good idea to optimize the primary and central metabolic pathway to improve the production of the terpenoids?
Yeah, I totally think it's a good idea. The more acetyl-CoA you produce, theoretically, the more beta-carotene you produce. And yeah, I think it's a valid approach. Also, like if you think the specific substrate you want to use, you can design also some pathways that directly convert your substrate into acetyl-CoA more efficiently, and then you can potentially maximize the process. I think depending on the organism you're working with and where the bottleneck on the pathway is the bottleneck. Normally, the bottleneck at the beginning is in the end. So if you're not producing a lot beta carotene, if you change many things upstream in the central carbon metabolism, I don't think you will get a lot in the end, because maybe your limitation is in the last steps and you accumulate something in the intermediate. But if you are already producing quite a lot, then it's very likely that the increase in the precursor will also increase the production. Yeah.
Okay, thank you.
Welcome.
Okay, Antonia.
Hello.
Hi.
Hey, thank you very much for that comprehensive talk. It was very nice. So my question is, when you were working on the beta carotene, especially with the promoters, were those promoters chosen specifically like to be good for the bioprocess, or were you just trying to maximize beta carotene? For example, like were the promoters using cheap inducers, or yeah, things like that?
Well, in this particular case, we wanted something that was constitutive, so we didn't need any inducer. So normally in bioproduction, like in large scale, you want to avoid inducers because they are normally expensive. Sometimes difficult to obtain in enough amounts. Sometimes they are even toxic. So it's better not to rely on inducible promoters. Or you can have auto-inducible promoters. These things like can be enhanced depending on the phase of the production. Like in stationary phase, they are induced, for example, but you don't need to add anything to the bioreactor. So yeah, in that case, we wanted constitutive. So we selected high expression promoters, medium expression, and low expression promoters to have a range of things.
So could you possibly have a higher beta carotene production with an inducible promoter?
In this case, no, because we don't know many inducible promoters for Gerobia, and the ones we know are not as strong as this one. If you have an inducible promoter that is stronger than a constitutive promoter, then yes, whatever is stronger will work. Also, that the inducible promoters are useful when you have something that is toxic that you don't want or is messing up with growth. You have a lot of metabolic burden, as we mentioned before, and your cell is not doing great growing. You can induce this later once the cells have grown. But in this particular case, because the cells were growing happily, there was no need for such approach.
Right, makes sense. Thank you.
Okay, Ayush, would you like to ask your questions?
Hello. Hello.
Hello.
Yeah. Hi. Thank you. Okay. So I have 3 questions actually. So the first one is that, like you explained in your talk, that the beta carotene production was higher in lipid-producing microbe than the other one, which was a strange thing. So while trying to automate strain development, how do you plan to deal with these kind of surprises that biology throws on us?
Well, I think this is a little bit the beauty of this, right? That we don't know everything because we don't know how it works. You find like surprises like this one, and that completely change the way you design your engineering. So they say here, and here is an example of different rounds of the DBTL cycle, right? You first do a round where you introduce this 3 gene, you expect something, and you suddenly find that the system is not working as you expected. And you learn why this is happening. And then you can adapt your second round of engineering. So I think when you find something like that, it's really nice because it first opens some basic research, so make us know more or how biology works. And the things that you learn from these organisms can probably be transferred to others, and sometimes even transferred to humans. So every knowledge is important, even for something that you don't know at the moment where you are finding it. But also for your particular approach, you now know that maybe lipids, if we further increase lipids, we maybe further increase beta-carotene, or maybe there is a sweet spot where the right amount of lipids and can maximize even more the production of beta-carotene.
Thank you. So the second question is that we know that the microbial growth is actually pretty slow when we're comparing it to the other kind of supply chains. And secondly, a part of the carbon that we provide to the microbe will always be utilized in the growth of the microbe. So these are some inherent inefficiencies in using living organisms. So are you trying to tackle these problems, or are you approaching these problems?
Yeah, so in a work that I didn't present here, we've been trying to also help the downstream part of the processes by, instead of keeping the lipids or the products inside the cells, to try to secrete them outside, so they facilitate the recovery and decrease the cost of the downstream process. So I think that it's kind of linked to this, because once you have something that can be secreted out and extracted, you can potentially have a continuous process where you only grow your microbes once, and then you keep them alive as a biological machine that is just producing but not growing.
Okay, yeah, makes sense. And the third question is, we know that CRISPR is not the best, and it kind of makes some mistakes and off-target effects edits. So while using CRISPR multiplexing, could you please explain more on the off-target effects that you've seen? And do you think that it would help to reduce them? Because if we have to do it only once, then the number of random edits would obviously go down.
Yeah, that's a good point. Yeah, so CRISPR is known to have target effects, and they can be critical, especially if you're doing gene editing and you are cutting the genome in unexpected places and integrating mutation. The kind of multiplexing we are doing is more in transcription and regulation, which means we overexpress or we downregulate transcription. And in that case, normally the off-target effects are not so critical. Normally you don't mess up with viability so much. I don't think it could happen. It could be, you could be targeting something that is important, an essential gene, something like that, but it's less likely that when you are editing and you can have problems on the viability of your strain just because you are making so many cuts in the genome, so you cannot, this repair system cannot work well enough to maintain cells alive. But there is a lot of research going on in decreasing of target effects, starting for the design of your guides.
Okay, thank you very much.
And Jakub?
Hello there, can you hear me?
Yes.
Excellent. It was an excellent talk. Thank you very much. I'm really intrigued about the 24 guide RNA CRISPR system. I would like to ask if you were able to achieve really those 24 or such a high number of edits in the yeast cell, because as far as I know, the problem with the multiplexing is that the system gets saturated quite quickly. And if you try to make more than 3 or 4 edits at the same time, the efficiency gets really low and you will end up with the population of cells completely differently edited. So how do you go around this system?
So yeah, it's kind of linked to the previous question, because the solution is the same. It's like we are using transcriptional repression. So we are not we are not editing genome, we are editing the expression.
All right, and then it works just right. If you are coupling it with the activators or repressors, then the CRISPR works well with 24 targets.
Yeah, I mean, it's not trivial because you need to be able to express them correctly and process them correctly, and this is what we are working on. But if you do it in the right way, yeah, it's possible and it's working for us. And the limitation is more in how do you express them and not in how many you express.
Sure, excellent. And do you think it will be able to do this system also in E. coli?
Yeah, I'm sure it is possible, yeah.
Great, thank you very much.
Mm-hmm.
Wonderful. Do we have any other kind of last questions, or is this it? Okay, so I think that this was it. I think that well, first let me thank Rodrigo for a really fascinating talk. I think that we learned a lot in a broad set of areas, and I think it was really great to learn about what all can be done in CRISPR in strain engineering and in metabolic engineering. And I guess that many of us got like really good thoughts about what we perhaps could work on further. So I would really like to thank Rodrigo for joining us tonight, and of course I'd like to thank Corina and Carolyn for organizing this talk. And finally, just a reminder: Synthetic Biology Society in Oxford is continuing, and next talk will be on Monday, July 20th at 1:30 p.m. with Dr. Nicola Patron from Oldham Institute, who is the leader for the synthetic biology in Norwich, and her research focuses on engineering photosynthetic organisms for industrial biotechnology and crops that are healthier to consume and less environmentally damaging to cultivate. So I really hope that you will be able to join us. Stay in touch with us on Facebook, Twitter, wherever. So thank you again very much. Carolyn, Corina, did I forget anything, or is that it?
I think that's fine. Thank you so much, Rodrigo, for giving the talk. We're super happy to have you.
Thank you. Yeah, it was a pleasure. If you have any further questions, you can email me or anything.
Great. So thank you very much, and have a lovely start of the summer break or holidays, or work duties or research duties, everyone. And we hope to see you in the next events.